Okay, let's get started, guys. Where do I start? So I uploaded the final exam today. There's 48 questions, not 50. The questions are very similar to the midterm, just a little bit changed in terms of wordings to avoid any misunderstanding. If there is a question, for example, like the interference one, I specify to read properly the question. There are some questions that are like some topics that are covered in 2 different questions. So I highly suggest you cover those. For example, the last questions of the midterm, you'll probably find it in the final. Okay. The gene in the middle and the distance in between. Okay. So study that. Yep. Yep. I'll give you the percentage in an email. Just have to make sure. What else? So the final exam will be for 3 hours in the MB building and in the second level below ground. Okay, that's where the metro is. So don't get lost. Go there a bit early. Bring your pen. No, not pen. Pencil. Bring a calculator, and you're allowed 1 sheet of formulas. Okay. The calculator has to be approved by the bookstore or some stuff like that with that little ticket there. Yeah. No, I will give you the formula sheet. Yeah, I'll give you the formula sheet because I feel like some people in the midterm did not do well just because they didn't remember the formulas, but they had the idea in mind, and we'll be learning some more formulas related to the Hardy-Weinberg equilibrium, which is population genetics. So some free questions and answers there. What else? So the calculators that you can use on the final have to be approved by, does anyone know what it is? I forgot what it was exactly. Exact, that code there. I forgot how to do it. It's been like 10 years. So I'll figure out how to do it with the department, and then I'll send an email for everyone, okay? The exam has questions that are in the type of the last 3 there, with the 1 question leading to the second one, third one, okay? There's a few like that, but they're not too complicated. There's a few questions that are just pure theory, like A, B, C, D, E, which one of these means this, okay? What protein in DNA replication does this kind of job? What else? What else? I'm trying to remember the exam. And your typical Punnett square calculation, frequency, all that stuff, okay? So that's all the information you're getting until the end of the semester. Now we focus on the course, okay? So there's a little bit change of plans. The first thing is that we're going to be doing proteins today. Thursday, we're going to start the lacopron and all that. I know in the schedule it's a little bit messed up, but I have to readjust it so that we can cover the whole semester. We'll be reaching the last chapter, which is genomics, so hopefully that will be okay. And then we'll have a day left at the end of the semester, the last week. So that's the week of the 8th of April. The Thursday, what I'm thinking of doing is a review session for the whole semester. And if that's not enough, and those who want to come, maybe I'll do one on the 16th also, which is the makeup day. Okay? So that should get you ready. Just make sure that you bring your pen for the pencil for the final. It's very important. Okay? Because I have to correct it quickly and submit it to the department for approval. Okay? With that being said, today's lecture is a bit packed, so I'm gonna go a little bit faster than usual, but we're gonna avoid a lot of the technicalities of protein translation because they're not really relevant for you to remember by heart. Cool. So let's take a bottom-up approach. We'll start by the molecular stuff and then we go to translation. So proteins, as you already know, are polymers that are composed of amino acids. So it's just a chain of amino acids. There's 4 different groups of amino acids, which we'll see in a second. You won't have to learn these by heart for this course, but probably in biochemistry you'll need to know all of them. Okay, so that's that. The amino acids, there's some of them that are non-polar, polar, charged and not charged, and then negatively and positively. So just remember the 4 different categories. That's going to be important for you. And so these amino acids have all the same, they all have the same structure. It looks like this. There is an amino group bound to a middle carbon, which is bound to a carboxyl group. The amino group is the N term, what we call the N term. And then the carboxyl group is called the C term. And then there's 2 other groups, two other sides. So there's this hydrogen here, and then this R. R is for reactive side chain. So this is what makes the identity of an amino acid, depending on what you have here. And they all have different properties. The first group are the non-polar amino acids. So there's a few of them. The only one that you need to really focus on in this course is methionine. Methionine is important because this is the first amino acid in literally every protein that exists, with a little modification for, for prokaryotes, but it's always methionine, okay? The others, not necessary to remember them for this course again, but you'll probably have to for biochem, so might as well start at some point, you know? There's also these polar uncharged proteins, amino acids. And what's important about these really is just that they can take up post-translational modifications. I will explain that in the last slides for this lecture, okay? Serine, threonine, tyrosine can get phosphorylated, and this usually activates or deactivates proteins. Cysteine get oxidized, so a bit of biochemistry, but cysteines get oxidized and they can break up the structure of a protein because of this SH group. So this thiol here, as we call it, builds disulfide bridges, which connects a protein together. So when it gets oxidized, it changes the structure of the protein, okay? These 2, forget about them for now. And there are polar amino acids that are positively charged and others that are negatively charged, okay? That's all the information you need to know. So the tyrosine, threonine, and serine. I was answering the question. So there's a peptide bond. So when these amino acids are built, right, they bind together. So one bond that is important for this here is called the peptide bond, which is a covalent bond that links a carboxyl terminal from one amino acid to the N terminal of the other amino acid, okay? So C term to N term. This is a covalent bond, and it happens by the loss of a water molecule. Cool. Very simple. What else? So there's a few bonds that we learned in this course, like phosphodiester bond, peptide bond. Remember those, okay? They're important. So proteins can be arranged in 4 different levels. The first level is the primary structure, where you have the amino end and the carboxyl end. Obviously, each amino acid has its own end, but the whole chain also has 2 polarities, the amino end and the carboxyl end, and it's just a chain of amino acids bound together. Carboxyl group to amino group, okay? By peptide bonds. And then the reactive chains basically alternate on each side. Secondary structure of the proteins is the alpha helix and the beta sheet. There are other types of secondary structures. So here I'm just showing this for you guys to actually get an idea about the possibility of the secondary secondary structures, but there are other types of structures which you will also learn in biochemistry, which is fully loaded, as you can see. It's a big course. So the secondary structure here that I'm talking about with the alpha helix is just, it looks like this, kind of like DNA, but for proteins. Okay, it's helical. And then the beta strand has 2 anti-parallel sheets that go in different directions and that interact with each other by hydrogen bonds. Okay. So if I heat up a protein a little bit, it's going to break the secondary structure, but not the peptide bond because it's a covalent bond. Okay, useless information, but you'll need it for the future. Tertiary structure, or third level structure, that's when the secondary structures come together. So you can have multiple alpha helices with multiple beta sheets, and they come together like this, and they the proteins start having these functional domains. For example, this is part of hemoglobin, and this is the heme group, which binds to iron. Okay, so this is the shape of the protein, and this, we call it a polypeptide. Now, hemoglobin is made up of multiple subunits of this unit here. Okay, and that's the quaternary structure. So when you have multiple subunits together, that is the full protein, and that's the quaternary structure. So you have two beta subunits and two alpha subunits, and they each bind an iron. This is hemoglobin. Cool. Is it simple up to now? Yeah. Okay. A lot of learning by heart. So now let's link it to actually the DNA, mRNA, and all that stuff. So there's a few features that link proteins to mRNA and DNA. And basically, your genetic code, the role for it is just to provide the blueprint for your cells to make the proteins. It's the proteins that are doing the big work in the cell, not the DNA. Okay. So these proteins, how do we get to them from RNA? Well, first of all, the RNA is divided into codons of 3 nucleotides. So 3 nucleotides each, and each codon, so this is the group of 3 nucleotides, will specify an amino acid. So each codon will code for an amino acid. Now, the linear sequence of nucleotides, so of the mRNA, is the same as the sequence for the amino acids in a protein. So when you read the mRNA from a 5' to 3', you'll see, from a 3' to 5', you'll see that you can build the protein from there. Okay, and we'll see this here. A few more important points before we get into the details. So the genetic code is degenerate. What that means is that it's not crazy, okay? It just means that the codons, some codons, so 2 different codons can code for the same amino acid, okay? Sometimes 3, sometimes 4, etc. So this is what we mean by the degeneracy of the genetic code. What else? The genetic code is non-overlapping. We'll see this in a bit. And it's continuous. That means when you start reading from one point, after that you read 3 by 3. 3 nucleotides by 3. Okay, and that's called the open reading frame. I'll show that in a bit on the board. But these are very important features of the genetic code that link the RNA to the protein. So first of all, if you remember, in mRNA we have these nucleotides here. So the sequence of them can determine which amino acids are built. And the way you read this is by looking at the first letter of the codon, and then the second letter of the codon, and then the third one, and it will tell you which amino acid you're making. For example, if the first letter is cytosine, and then uracil, and then whichever other third letter, then you're making leucine as an amino acid. For sure, in the final exam, you'll get the sequence that you'll need to transcribe and translate. Okay, write that down. So you need to be good with this, and you need to understand the directionality. Okay, everything gets synthesized from a 5' to 3'. Yep. Yeah, the same exact table is on the fine. I don't want you to remember all this stuff here. What I need you to remember is just that methionine is the start codon. So when you see this in the mRNA, the AUG, that means this is where translation starts. This is the first amino acid. You also need to remember these 3 stop codons. So whenever you see on the mRNA sequence UAA, UAG, or UGA, that means the protein sequence stops. So there's no more translation beyond that point. Cool? So these are the 4 things that you need to remember. The rest, pretty useless, except if you need to make a polypeptide, but usually you can find all this information online. It's just here that methionine, I mean, I remember it by heart now because we use it all the time in research, and these guys, same thing, right? But the rest I have no clue. I have to go back to the table still, okay? And these will be important, for example, if you're building your own plasmids, primers, etc., for research or whatever other purpose, okay? And for your fine. So you can divide it into blocks like this. This is just a different way to show the genetic code and its link to the amino acids. You can see that all these four here, they make serine. And this is just an example of the degeneracy of the code, because if you switch just the last code, the last nucleotide here from UCU to UCC, you still make serine, okay? Sometimes these changes will switch the amino acid, but most of the time they won't, and that's just the degeneracy of the code, okay? Make sense? Good. So the story about the overlapping and non-overlapping. So there are two possible theories of how the mRNA is read. You can read it either in a non-overlapping way, so three nucleotides and then 3 nucleotides, etc. Or what you can do is also read it in a different way. So you would read the first 3, and then your starting point will change to the second one, and then you read those 3. And you can also have the amino acids like this, okay? Many experiments were done to figure this out, but basically the result is that the code is non-overlapping. So it's read from a fixed starting point and then 3 units at a time. Cool. As you can see here, there's this AUG, that's the start codon. Methionine. What else? Did I forget something? Okay, now for the reading frame. This is an important concept that you have to understand. So the reading frame is where translation starts. So the beginning of a reading frame is where translation starts, and the end of it is when there's your stop codon. Okay, that's your polypeptide. So if you look at this here, for example, this was shown in the book for something different, but I'm using it for a different purpose here, just to show you the reading frame, okay? So if you have this sequence here like this, then no matter how you read the frame, you'll produce the same amino acid polypeptide, right? The only thing is that you won't have the last one. If you shift it to the left here, what's going to happen is that you'll shift the reading frame. You'll still produce phenylalanine, but one of them will be messed up because it will have only two nucleotides, which is not sufficient to make the amino acid. Same thing here. And then if you look at the example that I was describing, so let's say this is the first reading frame. So it starts from here and ends here. And you can see that you're making just multiple valine, right? If you shift it by 1 nucleotide to the right, what's going to happen is that you'll switch all the amino acids after that. And so you're producing here leucine, because these codons code for leucine. And then if you shift it another time, you'll make a different group of amino acids. Now, why is this relevant? Why am I explaining this? It's because in DNA, there's mutations that happen, okay? And there's a few different types of mutations. Substitutions is where you have just one base being changed for another. So when replication or transcription happens, there is a mismatch, and then you end up with a different nucleotide, and that could change the final amino acid. You can also have insertions and deletions. So for example, you could be deleting this C here, and it's going to mess up the sequence, right? Or inserting an additional C, because it's not a perfect process, okay? For example, where this is actually very problematic is when you have a switch in terms a substitution, single nucleotide substitution that leads to a stop codon. What happens with that is that you have your amino acid sequence and then suddenly it stops, right? And so you don't end up with a healthy protein, and that could be really problematic in terms of, pathways downstream and cause pretty much any disease, okay? There's a lot of diseases that happen just because of a single nucleotide mutation, cystic fibrosis, etc., okay? So this here is called a nonsense mutation. nonsense because it messes up the protein. And so it stops it when it's not supposed to stop. There's also another type called missense. So missense is when you have a change in one nucleotide, but it does not end up in a stop codon. Instead, what it leads to is a change in amino acid. Okay? For example, if you take a look at, say here, let's see. So if I change if I change the C at the end here to G, right? CAC, then instead it will become a glutamine. Yeah. So this is a missense when you go from one amino acid to the other just because of one change. The most dangerous mutations are either the nonsense or the indels, so insertion or deletion. Because if you have an insertion here in the middle with an additional nucleotide here, it will mess up all the sequence after it. Because the frame will, the open reading frame will shift by 1 or 2 or whatever. Yeah. The indel? The indel is just either insertions, so it's 2 words in one, right? So either an insertion or a deletion. Actually, I'll show you this here, maybe. Just quickly, and then we can continue. Okay, so let's say you have your AUG here, and you're trying to make a protein from this, and then you have, I don't know, AG. So if you have this sequence, you know that this is your start codon. You should learn it by heart. That's methionine, right? And then you have, I don't know what this one is, but let's just call it amino acid number 2, okay? So if I switch this G here to something else, for example, C, or I don't know, it's called a substitution, okay? And this substitution will affect the amino acid being generated. Because you're changing the start codon, you might not even code for this protein anymore, okay? So that's one of the dangers. The other one, that is called an indel. So you can have either an insertion here, for example, if I add another A, right? So this is an insertion. So now the reading frame is this here, that's the first codon. This is the second one. And then you end up with a free nucleotide here that's pretty much useless. Now this will not code for the same amino acid as AAG, okay? So that's another issue. This is the insertion. It's problematic for 2 things, because it shifts the frame, right? Changing all the amino acid sequence after, and on top of that, you end up with nucleotides that are pretty much useless. Another type of mutation is actually, there's no back function here. Okay, let's erase. A famous erasement. There's another type that is called the deletion. So let's say, for example, your transcription happens and you end up deleting that one. The deletion also will shift the frame, right? And then you end up with AG, and that's pretty much useless because it cannot make an amino acid. And obviously, indels are not just 1 nucleotide or 2. You can have indels of 2,000 nucleotides if you have to, okay? Just one important thing is that you have to read it in steps of 3. And the last one is when you have the stop codon here. So UGA, for example, and then you have, let's say, for example, a substitution here, and you end up with UCA. Well, this does not code for a stop codon, and so your synthesis will continue and will code even in the untranslated region. Yeah. Exact. So this will come, it will be sticked with this here. Yeah, exact. Cool. So I think that's pretty much it. Yep. So when you create a stop codon, it's called a nonsense. If you are removing the stop codon, there's not really a name. It's a missense, actually, because you're, that's good question. Is it called nonsense? This is actually a very good question. I have to Google this. I'm not sure. When you create a stop codon, it's called nonsense. When you remove it, I have to look it up. Coming with good questions here. Okay. What else? Let me see if I got the... Okay, yeah, not bad. I still have a good memory. Easy up to now? Simple? And by heart? Okay. Now let's get a bit, let's dive a little bit deeper into the translation. So now you know what, you know the different amino acids, you know how mRNA is converted into protein. Now we'll look at the machinery that actually does that job. And this is where it gets a little bit complicated. So focus, okay? So the main machinery that's important for translation is not a polymerase. It's called a ribosome, okay? Which is divided into two subunits, one called the small subunit, one called the large subunit, and it has a decoding center. This is where everything happens. Now this protein here is a mix-up of RNA, which you can see these little weird bubbles here, and proteins, okay? And we'll see the different types of proteins and RNAs. Actually the opposite. These white things here are the RNAs and these are the proteins. Okay, and we'll see the details of all of this in a second. The first component of the ribosome is the tRNA. If you remember last lecture, we talked about different types of RNAs. Today we'll be focusing on two main types, the ribosomal RNAs and the transfer RNAs. So transfer RNAs look like this. They're basically a single-stranded RNA molecule that goes from a 5' to a 3' direction. Few features that are important on this, the rest is useless. First of all, it has a clover leaf shape. I think you all know what a clover leaf is. With 4 double helical stems and 3 single-stranded loops. So this loop here, and this loop here. Okay. The shape here is important because it allows it to interact with the mRNA here and with the ribosome at other places. Okay. The second important feature is the let me cover this one first. So the second important feature is the amino acid attachment site. So these tRNAs, all they do is they come and bring an amino acid with them and then leave. Okay. And the amino acid attaches here at the 3' of the tRNA through this CCA nucleotide sequence. Okay. The third important feature and the most important is the anticodon. So the mRNA has a certain specific sequence, and each codon will code for an amino acid. So basically, this tRNA, what it does is it links the codon to the amino acid. Okay, and it has something called the anticodon. So let's say, for example, you have a codon which is, I don't know, one of these guys here, UCU, which codes for serine. So then the tRNA will have what? Will have AGA, and it will bring a serine with it. So the opposite of this is the anticodon. Okay. So this anticodon here will bind to the codon. Well, not bind to it, but interact with it through hydrogen bonds and bring the right amino acid to attach to it here. Make sense? Cool. A few things to remember about this tRNA. First of all, the CCA sequence at the end, at the 3' end, that's where the tRNAs will bind the amino acid. And the tRNAs have something a little bit special compared to normal RNAs. First, they are transcribed from the DNA using the 4 basic nucleotides, which are then converted to these special nucleotides. Okay? You don't really need to remember them by heart. Just know that they get converted. The nucleotides for tRNAs get converted. Okay? And virtually all tRNAs have the same shape. So this is something that is very well conserved. Actually, translation, transcription, and replication are very well conserved. Okay, they don't change much between different organisms. because they're super vital to the process, and it's very similar between organisms, especially translation. Okay, so tRNAs, how do they get made? So first of all, they come from transcription of the DNA. That's the first part. It will build this sequence here, the cloverleaf sequence. But you also need a step where you're attaching the right amino acid to it. So there's this enzyme called amino acyl tRNA synthetase, which basically all it does, it uses energy from ATP and whatever, and just attaches the right amino acid to the right tRNA. Okay. The specifics of this, no need to know it. I still don't know it, and it's probably going to be useless for you. But basically, just know that there's a protein, this protein here, that will take this tRNA and attach to it an amino acid based on the codon here. So there's probably some sort of codon here that has to be detected. And these amino acyl tRNAs, I'm pretty sure there are multiple copies, different copies for each tRNA. Cool. One thing that's important is that the amino acid is attached at the carboxy terminal to the 3' end of the tRNA. This here. Okay, this is where I lose a little bit of people. Hopefully not. So one thing that's important to know about the tRNAs is that, as I told you, the genetic code is degenerate. Okay, so that means that sometimes two codons will code for the same amino acid. It's the same thing for tRNAs. The anticodon, usually the first and second, nucleotide are always conserved in terms of interaction. So A only interacts with U, and G only interacts with C. But in the third one, it's different. In the third one can interact with, has different combinations, okay? For example, the third one, if it's a G, it can interact with a C or a U. And I showed you this in the transcription lecture. If it's a U, it can interact with an A or a G. And there's another type of nucleotide called inosine, or inosine, I don't know the way you pronounce it. But basically, this one can interact with A, C, and U. So you can have an I at the end. Okay, this is specific to tRNAs. So tRNAs don't have the same interaction mechanism between two nucleotides. They have specific base pairings. So because this third position here is kind of not always interacting like it's supposed to, we call it the wobble position. Okay? Yeah. Yeah, pretty much. But you don't need to remember the combinations. I'm just explaining the concept. Yeah. No, we are referring to the anticodon. Wait one sec. You see this? Oh, yeah, you're right. Yeah. So this here is supposed to be like this. Better? Yep. No. I'm not about memorizing things by heart. This is useless information. Okay. It's just to explain this concept, because the tRNA, as I told you, does not follow the same principles we learned for transcription. So as I just said earlier here, it is transcribed with the same 4 nucleotides. So adenine, uracil, guanine, cytosine. But then it gets altered. Those nucleotides get changed to dihydrouridine, pseudouridine, and inosine. And so it has different nucleotides. That's the idea behind this. And because it has different nucleotides, it ends up having this wobble-based pairing at the third nucleotide of the anticodon, which is at the 5' end of the anticodon, and these are the base pairing, but you don't need to learn them by heart. This is just the idea. Okay, so if I ask you on the exam something about this, I will give you this table. Okay, the rest is pretty much the same thing. Yeah, pretty much. Okay, back to these ribosomes. So we learned about the tRNAs, which bring the amino acid to the ribosome, and the ribosome is the protein, the big protein with the RNA that interacts with your mRNA. And so it has two subunits. In bacteria, there's the 70S ribosome, which is built from the 50S big subunit and the 30S big subunit. Now 50 plus 30 is not 80. Don't make the mistake on the exam. Okay. It's not an addition here. Okay. In eukaryotes, you have the 60S subunit and the 40S subunit, but they make the 80S subunit. Okay. So don't mix up these two. 50 plus 30 gives the 70, and 60 plus 40 gives the 80. Okay. Yeah, because it's not linear. So it's not a linear thing. So this is because there's a unit called the Svedberg unit, which is just the sedimentation coefficient. So when you run it in a centrifuge in a gradient, there's a coefficient that you can calculate, and it basically comes up to these numbers. Okay, it's not in terms of weight or whatever. It's more in terms of density and composition and all that. And you can see that the ribosomes have these two subunits, right, which are very important, and they're each made up of two components. So either a ribosomal, not either, both a ribosomal RNA and certain proteins. Now, I don't need you to remember the 23S ribosomal RNA and the 5S, etc. Okay, just remember that there's a big subunit, small subunit. Don't mix up the numbers here. And remember that the components of each of these subunits is the ribosomal RNA and proteins. Okay, simple. Because this is super complex, and if you start like learning it all by heart, there's no point, right? So that's a mistake right here. Let's fix it right now. So ribosomal RNAs are super weird. They look like this. They have a lot of secondary structures, and each specific ribosomal RNA is different. One thing that is important in these ribosomal RNAs is that they are the ones that connect the mRNA to the ribosome through this sequence here, which is called the Shine-Dalgarno sequence. So this is for prokaryotes, and I'll show you this specifically in a second. But basically, this sequence here recognizes an mRNA from prokaryotes, and the ribosomal RNA is itself bound to the ribosome. Cool? So this is like kind of a scaffolding RNA. That's that. And they're also important for protein synthesis because they catalyze the reaction. They have catalytic functions, and we'll see that in a second. Damn, going too fast, my head hurts. Okay. Next. Do you have any questions for me first before I go? Done? Okay. So back to this ribosome guy. There's two subunits. If you look here specifically, we're looking at the 50S and the 30S. So this is a prokaryotic ribosome. The 50S is the big subunit. And then you have the decoding center here in the middle, which has three compartments: the A site, the P site, and the E site. And this I will explain to you what it is right now, but you need to remember the names. It's important. So the amino acyl tRNA binding site, the peptidyl site, and the exit site. And you'll see why they are important. There's also another important concept is that the peptidyl transferase center, so this is the center where there's a transfer of the polypeptide chain being built. It happens in the large subunit. So most of the stuff happens in the small subunit except the peptidyl transferase center, which happens in the large subunit. Okay. And so the mRNA comes up Yeah. So the mRNA comes up this way. No, actually, no. The mRNA, so the ribosome attaches to the mRNA and starts moving along this sequence here. And as it moves, it will add amino acids to the polypeptide chain based on the sequence that it finds here. Okay. This is what it looks like. So you have your mRNA, you have the ribosome right here, which has the big subunit and small subunit. And then as it moves along the sequence from the 5' to 3' end, it will uncover new codons, right? So the codon will first be exposed to the A center, and then to the P center, and then to the E center, okay? And I'll show you the details about this, what's gonna happen. The other thing that's important structurally speaking is the peptidyl transferase center. So this is where the polypeptide that you can see here that is forming will be removed from here and added to this one. Let me just put my laser. So it will be moved from here and added on top of this one here. And then this tRNA will move to the side. Is it clear? Yeah? I feel like I'm going too fast. Okay, good. So back to this Shandal-Garneau sequence that I was talking about. Yeah. So these tRNAs are moving from this center to this center to this center and then out, okay? So what's going to happen is that this sequence here, the whole peptide will be transferred on top of this one, and this tRNA will go to the exit center and then leave. But the first one here, which has only one, now will move to the center, and it will have the change stays in the center. Easier way to say that. Good. That's pretty much it. Okay. What else? The peptide is added at the C term, as you can see. So this is the end term. This is the beginning of the peptide. Yep. Right. So most of this stuff happens in the small subunit, the decoding center, what we call the decoding center. The transferase center is happening in the large subunit. You can see it here, right? The big bubble here. The transferase center is here, while the decoding happens inside the small subunit. Cool. Yes, Susanna. Oh, yep. So I'll get into the details of this in a bit, but what happens is that, how do I show this without making it too complicated? So there's the tRNA that starts here. So so you have your mRNA sequence, okay, which goes from a 5' to 3', right? So the ribosome will attach at the first codon of the mRNA, okay? And then it will start moving from left to right in this direction. So as it moves, it will discover new codons, right? So the first time that the codon is exposed, it's exposed to the A site, right? And then the tRNA will come inside here, bring with it the amino acid, and then pass on to the P site, and then to the E site, okay? And I'll show the details of this. But basically, as this, yeah. Right, so imagine this here, move it right here. And then it starts moving this way. Make sense? No? Yeah, exactly. So it basically in this example, it would have seen this one first. That one would have been in the A center, and then, right? Exact. But I'll show you that in a second. I'm getting more into details of this. Okay. Cool. So translation. Remember I told you that the mRNA, the mRNA sequence actually has more than just the start codon and the stop codon. It also has those untranslated regions, which is the 5' untranslated region and the 3' untranslated region. So for translation to happen, it does start at the start codon, but before it starts translating, it needs a sequence before the start codon, which in prokaryotes is called the Shine-Dalgarno sequence, which is this sequence here. And this is important because it's this sequence that gets recognized by the ribosomal RNA, and then which is attached to the ribosome. Okay. In eukaryotes, there's something similar to this. It's called the Kozak sequence. Just remember that it's not just the start codon. There's a sequence before that that is important, and their names. The rest you can probably Google. Cool. Good. Okay, now the details. So before I get into the details, you don't need to remember the proteins that are involved in this whole process in terms of eukaryotic initiation factors and whatever. Well, in this case, it's the initiation factors. You don't need to remember them. Okay, that's useless because there's so many of them, and it's very complicated to learn by heart. What you need to just understand is that translation has 3 different steps, just like anything else. DNA replication has initiation, elongation, termination. Transcription, initiation, elongation, termination. So same thing for translation, right? So it's divided into 3 steps, and there are 2 different processes that are a little bit different between bacteria and eukaryotic cells, so our own cells. What's important to know is that the first thing that binds to the mRNA in terms of bacteria is called the 30S subunit, right? And it comes with a group of other proteins which we don't care about. The AUG, which is the start codon, binds to the P site, not the A site, the P site, which allows for the A site to have the first codon, the first codon of the thing, of the protein. Okay. And then what else? And then the ribosomal RNA that you can see right here binds to the Shine-Dalgarno sequence that I told you about, and it's also bound to the 30S subunit. Cool. So that's the initiation, and then there's these tRNAs that come to the ribosome and recruit the 50S subunit, and then it just starts moving along. Okay. Yep. About what? To the A subunit. To the A site, you mean? The A site. Okay. So the first thing that binds to the P site is the start codon. So if that binds there, that means the next 3 nucleotides are going to be in the A site, right? And that's your first amino acid, like after the start codon. Make sense? Yeah, that's pretty much it. No, it still does it. It's just I don't want to get into the details because there's a specific tRNA that is a bit different that brings the amino acid, the first amino acid with it. So just know that the start codon binds to the P, and then it aligns the second codon in the A subunit. Okay. So this is for bacteria, right? And it requires energy, usually in the form of GTP. Yeah. Let me draw it. Let's try this. So, but it was not working. Okay. So imagine the mRNA sequence like this, and then you have the 5' untranslated region, and then your first AUG is here. So when the ribosome comes, it will bind like this. Let's call this my beautiful ribosome. Okay. The AUG is the first codon. Okay. So it will bind at the P site, and then there's another codon here, let's say CCA or whatever, that will bind to the A site. Okay. The AUG, there's a special tRNA that will come and bind here, and it will bring with it the amino acid, right? And then the CCA, there's another tRNA that will come here and bind here. Sorry. And then there will be the transfer that I told you about. Okay? Does that make sense? Simple. That's all you need to know for this stuff. Okay. For eukaryotes, yep. Here? This here? Okay. So in prokaryotic mRNA, there is a sequence called the Shine-Dalgarno sequence. That sequence is bound by ribosomal RNA, which is part of the ribosome. Because, you know, this thing here needs to attach to the ribosome. Okay? And it needs to be aligned in a proper way with the ribosome. So the Shine-Dalgarno sequence is upstream of the AUG, which is the start codon, and it's bound by the ribosomal RNA to align the ribosome in the right reading frame. Does that make sense? Kind of. Okay. Yeah, it's a lot of for eukaryotic cells. It's a bit different because you have these eukaryotic initiation factors. So there's tons of these proteins from A to, I don't know, like G or something like this. And different ones bind different places. For example, if you remember the 5' cap, well, it's bound by these eukaryotic initiation factors, and they're important to align the ribosome with the mRNA. That's just that. That's all you need to know. You don't need to know the specifics of these eukaryotic initiation factors. The guy that discovered most of these eukaryotic initiation factors is at McGill, probably one of the next Nobel Prize winners. His name is Nahum Sonnenberg. So all this stuff that you're reading here is like top guy, okay? Still working. I think he's like 80 or 90, but killing it in research. Like he's working for like 6-7 people by himself. And so he discovered all this stuff here. I would not be surprised if you see him winning the Nobel Prize at some point. Okay, so this stuff here is all from him. Could have made it easier, but yeah. One second. The eukaryotic initiation factors. So it's a group of proteins. There's a lot of them. You can see here. There's EIF5, 2, 4. So each one of them has a specific binding site. I just wanted to focus on this part here, which binds to the 5' cap that is on the eukaryotic mRNA. That's it. Yes, Joe. Huh? Right. So very simple. The mRNA from prokaryotes, right? It does not have that cap, right? So that's the first thing. So there's a different way of aligning the ribosome, because there's this Shine-Dalgarno sequence that binds the ribosomal RNA and aligns the start site here. In prokaryotes, it's a lot more complex. There are these factors called the eukaryotic initiation factors that each bind different places in the structure. It's just to show you the complexity. And I wanted to focus on the fact that the 5' cap is something that is important for eukaryotic translation because it helps align the ribosome with the mRNA. That's it. The rest is useless for this course. Okay. There's a few things that come in play. The start codon, the Kozak sequence. In this case, I'm focusing on the 5' cap. Yeah. But the others are also important, you know. Yeah. The mRNA or the ribosomal RNA? Oh, which part? Sorry? The initiation factors? Yes. Okay. Where do I start? Let me think. Okay, so in eukaryotic mRNA, Remember, I was talking in the transcription lecture, I was saying that the mRNA gets modified by addition of a 5' cap and a poly A tail. Okay, so that's when the mRNA is made. So how is that relevant and why is it important? One of the functions of the 5' cap is for it to bind these eukaryotic initiation factors, which are just proteins that help align the ribosome to have that AUG inside the P site. Cool? Yeah, this is where stuff gets a little bit more complicated. Again, you don't need to remember the names of the proteins, just the fact that the cap binds some sort of proteins which help it align the ribosome along the mRNA strand. That's it. Maybe I'll make simpler for next time I teach this. I'll make simpler diagrams. Okay, and then elongation. Let's see, do we have time? Yep, so elongation. So now that initiation happens, so here we've showed initiation for bacteria. Here we showed for eukaryotes. And now we're going to talk about elongation, right? And this is in bacteria. And we're not going to focus on eukaryotes anymore because elongation and termination for eukaryotes is crazy. So we're just going to do the bacteria one. Cool. The reason why I showed this here, again, just because of 5' cap. So in elongation, you start with something like this, okay, where you have your AUG at the P site, and then there's this complex of another different protein that brings the tRNA to the A site for the second amino acid in the chain. Make sense here? And then there's a transfer of this chain here to here, and then this blue tRNA will come to the E site. This green tRNA will go to the A site and will contain two different amino acids, okay? Emptying the A site, which looks pretty much like this. The blue one will go to the E site, the green one will go to the P site, and then it will leave this one here empty. When that's empty, there's a third protein another protein that will come and block this site to give time for the tRNA in the E site to exit the ribosome. Yeah. By the way, I know this is a lot, but it's important to get into this kind of thinking because advanced biology classes, it's like protein after protein, and so it's not very important for this course. So I'm not going too much into detail. It's just important to understand how a gene is transcribed and translated. But the machinery itself, not so much, okay? But you'll be for sure learning this in other courses with the specifics of all these reactions, okay? So get ready for that. So this is for prokaryotic. For eukaryotes, there's another group of eukaryotic factors that are used for this function here. So EF1A, not important to know, but just to tell you, EF1A, which will replace this EFTU, and EF2, which will replace this EFG. Okay. Good. So then, when this tRNA has exited, the EFG leaves. This protein here that blocks the A site leaves, and that will allow for another EF2 to bring another tRNA for the third amino acid, etc. So it's a cycle. Easy? No? Sorry? Yeah. So it's empty like this. There's a tRNA that comes here, and then they will interact with each other. There's a transfer of amino acid, and then they move to the left. And so this tRNA will end up in the E site, the other one will end up in the P site, and the A site will be blocked for the time it takes to this empty, uncharged tRNA to leave the E site. And then it just keeps happening. I think if you have the book, there's probably videos on this. I don't know who's reading the book here, but I think there are videos on this. So you can just watch the video, not to learn the details, but see it actually rotating. Okay. The last step, the termination. Again, this is for bacteria. Eukaryotes, it's a bit more complicated. The stop codon does not bind a tRNA. This is the only difference I need you to know. The stop codon binds a group of proteins called the RF proteins, which will, because there's no amino acid, right? The stop codon does not code for any amino acid. So the termination happens through the binding of these RF1 and RF3 proteins, which recognize the stop codon and just disassemble the whole ribosome. Cool. There's a release of the peptide when these two bind, and then the ribosome ends up just detaching with the help of different factors. Yep. Just a protein. It's not an RNA. Oof, we made it. Okay, this is probably the hardest lecture, okay? The most packed, dense lecture. After that, it's going to go back to the usual rhythm, okay? So when we talk about, we're learning all this stuff here for what? To understand how inheritance happens, right? And so inheritance is just what genes are expressed and what genes are not expressed. And so there's different ways to regulate the amount of genes being expressed. For example, through increased transcription. So if you have a DNA, a gene that is transcribed a lot more than others because of activators or enhancers or whatever, then that's one way to express more of that protein. For example, if it's a pigmentation protein, like for the flower, then you would have increased transcription and increased translation, leading to more pigment. There are other ways to control the levels of the expression of a gene, because really it's not just like the Mendel stuff we saw at the beginning, right? It's not just you have the gene, that means you end up expressing it. It's more kind of a continuous thing, right? Where you have different levels of expression of proteins, and that will determine actually the phenotype. For example, the flower color, you can have mutations that make it transcribe more or less, and then you would end up with darker or lighter pigments. Cool. There are other ways to change the expression of a protein or its activity is through post-translational modifications. So when the protein comes out, just like the mRNA, it gets modified. And there are different modifications that happen. You don't need to know them by heart. It's just to show you all the different types. Usually you probably know about phosphorylation. You probably know about ubiquitination, which leads to the breakdown of the protein. So if you have a lot of ubiquitination in a protein for pigment, then you'll end up degrading it. So even though you have that allele and you have that gene, doesn't mean that you'll have that phenotype. This is why it's super complex, okay? So ubiquitination affects the level of the protein. If you have, for example, that cysteine that I talked about, and there's a disulfide bond, so two cysteines bound to each other, and then you break that can affect the activity of the protein, changing its expression and changing the phenotype, okay? This is just to give you an overview of this whole thing here. It's not just what gene you have, but also what happens with the gene, the mRNA, the protein, etc. Cool? So that's pretty much it. And there are other modifications. For example, how that protein gets localized. So if you have a protein that is necessary to be in the nucleus because it helps in transcription, and then it has a tag that localizes it, I don't know, in the cytosol, then it's not doing its job. Even though the protein is expressed, the mRNA is transcribed, DNA is replicated, you still have the phenotype of the mutant and not the wild type, which is supposed to be in the nucleus. Okay? It's super complex, so it's not just how Mendel saw it the first time. It's actually crazy. Okay? What else? More examples of post-translational modifications that I'm talking about. Yeah, that's pretty much it. So there's that. And the last slide, not bad. Super fast, but not bad. I feel like I talk too fast, but I'll take your questions. So I'll tell you a bit about my research now that you understand this whole concept. So there's a group that did research on a group of female patients that had breast cancer. Usually, if you look at the hormonal levels, females don't have, or women don't have androgen. They don't have a lot of androgen. So there's a group of women that have a lot of androgen. For example, if you ever heard of the polycystic ovary syndrome, usually it's caused because you have too much androgen in the body. And so what that does is that it leads to breast cancer. And it leads to one of the most aggressive breast cancers that has no cure. Cool. So what they did, when they took the DNA from all these patients and sequenced it, they found that there's one gene that has a lot of mutations. Okay, this is my gene of interest. It's called the estrogen-related receptor. And they found that most patients had these modifications in this gene, right? So R352P means that we're changing the amino acid arginine at position 352 to a proline. You'll understand the implication of this in biochemistry, but this thing here affects the structure of the protein. So now I want to study this. How do I do it? Do I go to the patients and start inducing these mutations? No, I can't do that, right? So I have to do it in mice and in cells. So what I do is take the sequence of this gene, go to the nucleotide that I can change. So I mutate that nucleotide for it to switch from arginine to proline. So if we go, just a little bit of interesting information. So where's the arginine? It's right here. And then proline is right here. So how can I change this? CCA, is there another arginine? I forgot exactly which one it is, but let's find it. Right here? Oh, there you go. Okay. So I forgot exactly which one it is, but just to give you an example. So let's say, for example, my codon was this one here, CGU. Then for me, I would just mutate the G to make it CCU, and that will affect the protein being synthesized. So that will give me kind of this not chimeric, but mutated protein. And then I do the same for the other two, arginine to leucine and leucine to proline, okay? And then I just make this mutant protein and use it to, and I basically change the one inside the cells that I'm studying or the mouse to this type of protein. And that allows me to study it in a physiological state. Yep. Sorry? Right. So usually the whole genome is sequenced. So I have the sequence of this protein or the DNA of it. And then all I do is I take the reading frame that I talked about. I find the start codon and then start reading from there the protein sequence until I get to position 352. So then 352, if I convert it into DNA, it's times 3, right? Because each amino acid has 3 nucleotides, and then I just find the right one there, okay? And then I mutate it using some techniques, and then followed by CRISPR inside the mouse or whatever, and then you can see that they start getting breast cancer. Not nice, but we have to do it to find a cure for this kind of stuff. So this is how the knowledge that you're learning right now is used in research at the moment. Yep. So PCOS has risks for cancer, not leads to cancer, okay? Because usually there's an imbalance in the hormonal state and it can get bad. Don't get scared. Everything can lead to cancer these days. So, but it's just an example of where you would study this kind of cancer. Okay, that's it. Questions, answers? No. Cool. So this is a very complicated lecture. Go over it a few times to remember everything. The next lecture. So Thursday we're gonna start the lacopron and all that. Cool. See you later, alligator.